Thank you, Cliff. Let me just start by saying that I live in Houston, an area frequently battered by hurricanes. So when Sandy made a beeline towards the New York, New Jersey coast, we watched with increasing concern. And we have watched with admiration and great hope that this great city will recover soon and will get back on its feet. So our thoughts are with you. My area of research is really a team research, and my major co-conspirator, Min-Chi Hung, is not here with me, so I'll have to impersonate him too. And what we have been working on over the past several years is to use the control mechanisms of the normal cell, which frequently go AWOL in a cancer cell, to encourage the cancer cell to commit suicide. Now, that sounds simple, but it's very complicated. So there are families of, in a normal cell, when some sort of injury happens, the cell takes a time out and either focuses on repairing that damage, or if that is not possible, there are signals and mechanisms and processes within normal cells that then force the cell to commit suicide. That's how we keep our bodies full of normal cells and eliminate the ones that are no longer normal. That's true for cells that start the process towards cancer. However, once the entire transformation has taken place, that control mechanism stops working well. Now, over the past several years, Min-Chi Hung has found a couple of genes that are in the family that encourage the cell to commit suicide. And he has transformed those by molecular biology procedures so as to give them much greater power. And in the lab, we have been able to show that by administering this to a collection of cancer cells, we enhance the cancer cell death rate substantially. Now, that works very well in the test tube. The challenge is, how do you make that happen in a human being? How do you deliver this internal poison, if you wish? And we've been working on mechanisms of delivery. This is a form of gene therapy, if you wish. And we have been using a variety of delivery vehicles, little fat droplets with molecular zip codes that deliver this family of genes. We have used viruses that, again, are attracted by cancer cell deposits, and a variety of other such delivery systems. Now, most of these are fairly inefficient, so that you can deliver to your targeted area only about 1% of what you want to deliver. So to enhance this, we have developed a couple of techniques that improve the specificity of the molecular zip code. The molecular zip code that finds its way to that area of the body where cancer cells are. So that's what our work has been focused on for the past couple of years. We are now ready to start the regulatory phase of our work. So we are out of the lab. We are starting to do the preclinical toxicology that the FDA requires in order to take this into human beings. We think we have a couple of delivery systems that are working very well. We have done and repeated the assays and the tests in laboratory animals. So we hope that within the next year or so, we will be starting our first clinical trial with this. This sounds complicated. It actually is. And we hope that it will work as well in humans, and it will be one of the magic ones that Cliff mentioned.
Great. Thank you very much, Gabe. I'm sure there'll be questions and discussion on that to follow.